All right, so I'm happy to introduce our next speaker is Susana Magadan from the University of Vigo and the University of New Mexico. Remote.
Hello.
Oh, we can hear you.
Ah, okay. Yes. Okay. Good morning. Good morning, everyone. I'm Susana Magadan, and first, I'd like to thank you to the AIRRRC community for inviting me to give this talk about establishing a standardized nomenclature for salmonid immunoglobulin and TCR genes. And of course, thank you for attending to the talk, even if it's online. I hope you enjoy it. So to start, I'd like to say a few words about the adaptive immune system in vertebrates. As you know, in your vertebrates, the adaptive immune system is based on clonally diverse B and T lymphocytes, which display antigen-specific recognition through immunoglobulins or antibodies in the case of B lymphocytes and T cell receptors in T lymphocytes. In addition to generate the antigen receptor diversity, the AIRRRC enzyme complex plays a main role in the rearrangement and recombination of VDJ genes to generate the receptor variable region.
Susana, excuse me?
Yes.
You're not sharing your presentation.
Sorry?
You're not sharing your presentation.
Ah, let me see. Okay. All right. Let me do again. And now?
Now, yes, good.
Okay, sorry. Sorry. If I do this. Okay. Oh. Okay. So, however, there are approximately 60,000 species in your vertebrates. We have mammals, amphibians, and birds, and teleosts, and we can expect difference between them. For example, if we compare teleosts and mammals, we can see that teleosts have immunoglobulins and TCR and have 8 and RAC enzyme complex. But for example, about the hypermutation, they generate hypermutation, but there is no affinity maturation, or the affinity maturation is very low compared with mammals. Or for example, there is no immunoglobulin class switch. And for example, related to the lymph organs, there is no exactly a bone marrow. They have a kind of what we name kineopronephros, that it is the organ who plays the role of bone marrow, and there is no lymph nodes or germinal centers. So maybe the lack of germinal centers and the follicular dendritic cells in teleost can an effect in the affinity maturation. About the immunoglobulins, we know that teleosts produce 3 classes of immunoglobulins: IgM, I have here the IgM, IgD, and IgT or IgZ. In general, or the last works suggest that the IgM play an important role in systemic immune response, and the IgT or IgZ play a main role in mucosal immune response. However, about the IgD, we don't have many information. About the immunoglobulin heavy chain locus structure is in translocon as in mammals. So we have different V genes, V and T genes followed by exons that codify for the constant region. But as you can see here, the CETO gene has its own DJ cluster, different than the DJ cluster or gene cluster that is related to the IgM and IgD. So it means that the production of IgM and IgT is generated by gene recombination and not by class switching. So let me say something else about the immunoglobulins. Before 2005, we thought that the teleost only produced IgM and IgD. But in 2005, John Hansen and colleagues identified a new immunoglobulin gene in rainbow trout, and they named IGT for teleost. In the same year, Danio and colleagues described a new immunoglobulin gene in zebrafish, and they named IGZ, maybe due to the zebrafish. Since that, a new sequence related to this IGT and IGZ have been discovered or identified in many teleost species, and many of them adopted the IGT name and others adopted the IGZ name, and mainly in those species related to the cyprinids. But if we do a phylogenetic analysis, we can see clearly that IGT and IGZ are evolutionary forms of the same antibody isotype. So after discussions with the zebrafish nomenclature committee, we now encourage a shift from IGZ to IGT nomenclature. Other important thing to mention about the adaptive immune response in teleost is that we know that the B cell and T cell development and immune response follow the clonal selection theory proposed by Barnett, and we can detect a specific long-term protection after vaccination. However, there are many open questions about or related to the adaptive immune response in teleost that we don't know. For example, we don't know how are the kinetics of adaptive immune response, or if the lack of germinal centers and lymph nodes have a dramatical consequence in the B-T cell biology or memory. Or, for example, we don't know exactly what is the basis of the humoral memory. We know in some species that there are a high stability of the antibodies, but we don't know exactly if the memory is more related to the long life lasting plasmatic cells or due to the presence of memory B cells. We didn't identify a clear subset of memory B cells. Or, for example, we don't know how do B cells participate to the mucosal versus systemic defense. And of course, we think that the characterization of adaptive immune receptor repertoire is a straightforward approach to address these questions. Even more if we think that we don't have this kind of tools like monoclonal antibodies to study the adaptive immune response. In fact, the first manuscript published where the deep sequencing was used to analyze the adaptive immune repertoire complexity was performed in 2009 using zebrafish. In this manuscript, the authors described the complexity of IgM in a full organism, but considering like the depth of the 454 sequencing and even the extraction of RNA and how they did the methodology, we cannot confirm that they analyzed the total immune repertoire in the organisms. Since that, few manuscripts have been published about applying the deep sequencing to analyze the immune repertoire in teleost. These are two examples where we use the 454 sequencing or the Illumina-MySeq technology to analyze the complex clonal IgM and IgT response in rainbow trout after systemic viral infection or after nasal vaccination with inactivated bacteria. However, the clonotype analysis in both articles or in both works were based only in V subgroups. We have different problems to overcome to do further analysis. For example, there is no full loci annotation, there is no standardized nomenclature for the different BJD genes. And of course, we have to take into account that fish is a very large group. So we can expect loci divergence, but we should keep in mind when we develop a nomenclature, the consistency between families when it's possible, and if it's not possible, at least within families. And considering this, our main objective is to review and update the description and annotation of the immunoglobulin and TCR loci present in Atlantic salmon and rainbow trout to build an unified or standardized nomenclature for salmonid species that will facilitate the analysis of AIRRSeq data. Well, we are working with the IMGT stack or IMGT system, as you will see. Maybe your first question is, why do we study salmonids, and in special the Atlantic salmon and rainbow trout? Well, there are different questions. One is that both species are very important for aquaculture industry, and both species have emerged as fish immunology models. Even more, if you ask a person who works in aquaculture about zebrafish, maybe he or she will answer that zebrafish is not a fish. So they consider that it's better to use the Atlantic salmon and rainbow trout because they are very important for aquaculture. And also both species are important from a biology and evolutionary point of view. Due to the anadromous migration in salmon, we know that salmon during the multiplication suffer a kind of immunosuppression, and also because there was a kind of whole genome duplication in the common ancestors of all salmonids. So we can study how this genome duplication affect to the diversity of the genes and the functionality, and of course, to study the effect on immunoglobulins and the TCR loci. So when we started with the annotation of the immunoglobulin heavy chain loci, there were two genome assemblies available for Atlantic salmon and rainbow trout. In the case of rainbow trout, this genome assembly was obtained from Swanson line, and they use the short read sequencing technology. So this genome has many gaps. So we decided to start with the analysis of Atlantic salmon. In addition, we had previous data, fantastic work made by Ben's Cook Group in Canada, like more than 10 years ago, where they characterized two immunoglobulin heavy chain loci in salmon, in Atlantic salmon, based on back sequencing. You can see here in this figure the two loci. They named one was named A and the other B. As you can see here, both loci are very large, more than 650 kilobases. Both of them have more than 150 B genes. And in both of them, we can distinguish several genes codifying for IGT, you can see here in red, one gene for IgM, and one gene codifying for delta. So based on this last landscape, we can think that to generate a complete and correct assembly or annotation for the salmonid immunoglobulin heavy chain loci is really complicated, not only due to the duplication of the loci, but also because there are many different V genes and subgroups, and also because there are a high number of pseudogenes. Anyway, we use this information to do the characterization of immunoglobulin loci in Atlantic salmon using this genome assembly. And we found the same two isoloci, the A and the B. A is in chromosome 6 in reverse orientation, and B is in chromosome 3 in forward orientation. About the names of constant, or about the constant gene names, we use a IMGT rule, and it's a very easy rule. So the first thing is that we add a D to those genes, or a D letter to those genes that are located in locus B. You can see here in this column the names of constant genes that are located in the locus B. In the case of IGT genes, to distinguish the different genes, we added a number that indicates the locus position. And the number 1 means the, it's better, the number 5 in this case means the closest gene to IgM gene. About the D and the J segments, well, you can see here that if we focus on IgM and IgD, these two IgMs, even if they are from the different locus, they should be expressed because they have a complete cluster of IgD and IgG genes, and there is no stop codons. But in the case of IGT genes, there are some genes without DJ cluster, like IGT1 in the locus A, or for example, the IGT2 in the locus A has many stop codons. So in theory, only one IGT per locus should be expressed. And related to the nomenclature, we follow the same rule as with the constant genes. And you can see here one example. In black, you have the gene group. Then we added a number related to the position of this D segment in the D cluster. Then in this case, we added T3, that it means that this D gene is associated to the T3 gene. And then we added the D because this gene is located in the locus B. Related to the B segments, we annotated less number of total sequence in the genome if we compare with the previous work did by Ben's Coop group. But we annotated more functional or open reading frame V genes. And we could cluster all these V genes in 16 subgroups based in the nucleotide identity higher than 75%. So then we use all these sequence to annotate or to characterize the immunoglobulin heavy chain loci in Rainbow trout using the Swanson genome. And of course, we found two loci, A and B. A is in the chromosome 13 and B in chromosome 12. Both of them are in forward orientation. And we found, in general, we found less sequence than in salmon. But for example, about these segments, they could cluster in the 16 subgroups that we identified previously in salmon salad. And for example, here in this phylogenetic tree, you can see how the functional and open reading frame B genes can cluster in 16 families. It's true that, subgroups. It's true that there is no branch for subgroup 13, but in this group, there are only pseudogenes, so it doesn't appear in this phylogenetic tree. Then we submitted all this sequence to IMGT, and the IMGT system generated the different reference directory sets. We show only the V genes that were included in the IMGT bequest reference directory. And for example, we included 135 genes, genes from Atlantic salmon and 77 from rainbow trout. Then we analyzed the functionality and the expression of this immunoglobulin using rainbow trout and using individuals from Swanson line. and using the nomenclature that we created and the IMGT system. So we generated different libraries for IgM using the spleen, and we could see that approximately 94% of the sequence could be associated to a gene, or could be identified the gene, the V gene. And also we could see that both loci can be expressed, or the V genes from both loci can be expressed. However, when we analyze sequence from other lines, not different from Swanson, these results are not so good. So the data set is not complete. But we were very lucky, because at the end of 2020, a new rainbow trout genome assembly was released. This genome was generated from another line, or another strain, is the AIRRSeq strain. And they use long-read sequencing technology. So there is no gaps, maybe few of them, but compared with the Swanson genome, this genome is really, really good. So we annotated the different genes, and again, we found less V genes if we compare with the data obtained from Swanson line, but we got more functional and open reading frame genes. And again, all of them can cluster with the 16 subgroups identified previously. However, when we compare the locus between both strains, or the loci, sorry, we can see difference, and mainly in the locus B. In these figures, we show the map of.
I just want to give a 5-minute warning so we can have time for questions.
Why? Okay, sorry. So the thing is that there are genes that we only found in Swanson, and genes that we only found in AIRRSeq. And maybe these genes that are only found in AIRRSeq can be a problem for a nomenclature based on localization, or based on the position of the gene. But we solved this situation adding a number in the new genes. So you can see here how is the nomenclature. And this number is only related to the date of the gene description. So it's not a positional number, okay? So we keep 14th in this case, because it means that these 3 genes are close to the pseudogene 1D14 that was found in AIRRSeq and in Swanson. So we included all this data in IMGT database, and AIRRSeq has been added as an alternative. So now we can analyze Swanson or AIRRSeq. And then in 2 minutes, I'm going to tell you something about the TCR alpha beta. And I go further in the presentation because, okay, the only thing is to show you that, okay, we annotated the TCR alpha locus. We found a high number of V and J segments, more in Atlantic salmon. These V segments can be classified or clustered in 26 families. In general, the distribution of the members, or the members in the different groups is similar if we consider members from salmon, salad, and rainbow trout. There are some difference. For example, there is one subgroup with only 1 gene in AIRRSeq, or the group 3 with 7 genes from Atlantic salmon and 12 from AIRRSeq, and the group 13 with 6 genes and from Atlantic salmon and 1 in AIRRSeq. And there is a region only present in a salmon salad from the locus. These 29 segments are only present in Swanson, and in this region there is no gap, so there is no these segments, but the rest of the locus are very similar. So we can adopt the annotation, the positional annotation. There are atypical TRVs, for example, TRVs belonging to the 3 subgroup without CDR2, and maybe it could affect to the MHC interaction. And the analysis of the deep sequencing libraries show that all these segments are expressed except in Atlantic salmon, except those belonging to the subgroup 4. And also the TRG gene expression, all of them are expressed. And unlike described in many mammals, no clear pattern of preferential VJ combination could be determined in Atlantic salmon based on localization. And we analyze also the productive and non-productive sequence. So to summarize, we have established a consistent and standardized IMGT-based nomenclature that can be updated and adapted to new assemblies of other strains or species within Salmonids. The Salmonid immunoglobulin heavy chain gene reference dataset is currently implemented in IMGT high bequest and IMGT gene tables, but further work is required to implement the Salmonid TR alpha and delta gene reference dataset. Of course, we have more work in progress. We are analyzing new genome assemblies related to the Salmonid. We are checking the polymorphisms, and now we are currently working on Salmonid TRB loci, and we can say that this IMGT-based nomenclature can be also adapted. And I'm sorry, but I cannot finish without the acknowledgements. I want to say thanks to my group, and especially to Africa Gonzalez, that is the PI of the group, because my group is mainly focused in human immunotherapy, but they are always open to these crazy research lines. I want to say thank Marie-Paul for all the discussions, to the IMGT system, to Pierre Boudinot, of course, because he's really my mentor in this field, to Alexei Krasnov, Eva Steen, and Uni Greenhold for all data in Atlantic salmon, to Benku, because even if he's retired, he's always open to discuss, to the AIR community, not only for the invitation, but also for the germline database working group, because I know that I'm not the most talkative or participative in the meetings, but you have to know that you are supporting and pushing our research. And INIF party for sharing the genome from Arlie, Oriol, Irene, and John Hudson. And that's all. And thank you very much. So it's time for questions. I think that I'm going to put the camera on. Okay.
All right, I think we have time for one question.
Hi, Susanna, great talk.
Hi.
Were those back clones that Coop used, a long time ago? Are those still available by any chance, or more, more long range sequencing?
So sorry, you asked me if I know if the backs are available?
Yeah.
I don't know. I don't know. I suppose that the Benku groups is keeping the bags in the fridge. You see, we don't know. We use the information in the manuscripts and the annotation that they did.
Yeah, I was just wondering if they might, provide any, regions that are not fully assembled or something like that. Yeah,
but we didn't work with that. We are waiting for the genomes or the new genomes that will be published. And for example, from Atlantic salmon, there is a new reference genome, so we have to check again. Okay, but you could see in the presentation that the data that we got is from 2018, and we have to recheck and check and check again.
Thank you.